All right, welcome everybody. My name is Rob Mettleman. I am a postdoc at St. Jude Children's Research Hospital, and I'll be moderating today's ATS Journal Club. So I'm really excited to welcome you all to this ATS Journal Club sponsored by the section on genetics and genomics. Today we will be discussing the article entitled An Aberrant Immune Epithelial Progenitor Niche Drives Viral Lung Sequelae, which was recently published in Nature this past September. The journal club is being recorded and will be posted via a link on the ATS website. So if you have colleagues who had to miss this session, this will be available there. So presenting the article today, we're really excited that one of the first authors on the study, Dr. Harish Narasimhan, from the Department of Microbiology, Immunology, and Cancer Biology at the University of Virginia. We're also excited to welcome several panelists for the discussion portion afterwards. We would like to welcome the corresponding author on the study, Dr. Ji Sun, who is a professor of medicine in the Department of Microbiology, Immunology, and Cancer Biology at the University of Virginia. We have Dr. Angela Rogers, who's an associate professor of medicine in the Division of Pulmonary and Critical Care Medicine at Stanford. We have Dr. Charles De La Cruz, who is a professor of medicine and director of the Acute Lung Injury and Infection Center in the Department of Medicine at University of Pittsburgh. And lastly, we have Dr. Joseph Misgerd, who's a professor of medicine and director of the Pulmonary Center at Boston University. So welcome all. So what we'll do is we'll start with our journal club with a presentation by Dr. Narasimhan, and then move to a question and answer discussion. So if you have questions, please add them to the chat. So we'd like this to be kind of a lively discussion between audience and panelists if possible. So if you're joining us online and like to pose a question, like I said, please type it in the chat, and then either Katie or I will use the webinar interface to introduce those into the discussion. All right, so now I'd like to invite our presenter to share their work with us, and go ahead and unmute and take it away. Thanks.
Great. Thank you so much for the introduction, Rob, and for this opportunity to share our work with you all. As Rob mentioned, I'm actually a graduate student. I will be defending next month, so I appreciate the vote of confidence. But I am very excited to share this work with you, what we've been doing over the last couple of years in the Sun Lab at UVA. So in the lab, we've been interested in this idea of chronic disease after viral infections for a while now. And this really came to the fore over the course of the COVID-19 pandemic through this phenomenon called post-acute sequelae of SARS-CoV-2, which I'm sure a lot of us are familiar with. It's essentially when individuals go on to develop a variety of impairments in potentially different organ systems. And this typically occurs well after the period of acute disease when, in fact, you cannot isolate infectious virus from these individuals. Now, given our lab's interests, we chose to focus on impairments or deficits of the respiratory tract. So people have reported shortness of breath, cough, and sometimes required oxygen supplementation as well. And just as a quick side note before I proceed, I'd like to make the case that this phenomenon is not unique to SARS-CoV-2. It's actually been reported after several other respiratory viral infections, SARS-CoV-1, MERS, influenza, RSV, etc. I think it's the scale of the COVID-19 pandemic that's really made this very obvious and apparent, but I think it's an important question for the field to keep in mind when thinking of these infectious agents and potential pandemics. And so in addition to the shortness of breath and cough and things like that, there can be a subset of individuals that actually develop a more serious manifestation where they go on to develop pulmonary fibrosis after COVID. And this has been termed PASC pulmonary fibrosis or PASC-PF. This is just an excerpt from the UK ILD study, where really what I'd like to draw your attention to is this column here, which, based on the criteria that was used for the analyses, you can see anywhere from 7 to 11% of the cohort was found to exhibit lung abnormalities up to 8 months post-infection. And this is when that interim analysis was conducted. And in independent reports, this has been observed up to 3 years post-infection. So this is not necessarily something that happens only very acutely, but can actually be a very protracted response and something that you see up to an extended period of time. Now, the cellular and molecular etiology of this condition was largely unknown, and as a result, we really don't have any definitive therapies to specifically manage this condition. And so our lab has been interested in this for a while now. And a few years ago, we published a study where we looked at lung function in COVID-19 convalescents. And these were individuals that were about 2 to 3 months post recovery and or post-infection. And you can see when you look at the lung function with respect to the cellular composition in their bowel fluid, we see that the lung function negatively correlates with the abundance of CD8 T cells within these people. And so what this suggested at the time was that perhaps the persistence of these CD8 T cells in these post-viral lungs might potentially contribute to chronic sequelae. And so the question still remained how this might be happening. And while we've tried to address this in several different facets, the bulk of my thesis work revolved around understanding whether this happened via the regulation of repair and alveolar regeneration. And so I'm just going to give a very quick primer about the process of alveolar regeneration. I'm sure most of you are familiar with it already, but since I'll be mentioning these terms a lot, I wanted to introduce them. So there's a very simple schematic of the alveolus, specifically looking at the epithelium. So we have alveolar epithelial type 1 or AT1 cells and AT2 cells that primarily make up this space. During the course of a viral infection, either due to direct infection or consequent immunopathology, there's substantial AT1 and AT2 cell loss, which, given how integral they are for the gas exchange function and just to maintain homeostasis in the lung, I'm sure you can appreciate these need to be replenished at the earliest to maintain optimal lung function. And we're quite efficient at doing so. This is primarily mediated in this environment by AT2 cells themselves. They're very efficient at self-renewal when they detect a loss in the surrounding environment. And not only do they replenish themselves, they can also transdifferentiate into AT1 cells via this transitional state characterized by very high expression of cytokeratin 8 or KRT8. And this is what happens the vast majority of the time we have an immunological insult or alveolar injury. But in a subset of cases and in a subset of individuals, for reasons that aren't fully understood, some people develop what's been termed dysplastic repair. And this is when these transitional cells tend to accumulate in this undifferentiated state rather than differentiating into a mature AT1 cell. Not only that, there's also been reports of the recruitment of KRT5 positive basal cells from the upper airways in the context of severe injury, which do a very important job of maintaining the structural integrity in that acute phase of infection. But when they stick around for an extended period of time, they're in fact biased towards becoming or adopting upper airway cell fates. So having those kinds of cells in the alveolar space causes what's been termed bronchialization of the alveolar epithelium, and again, can be detrimental when thinking of lung function and recovery. So for the purposes of this talk and like this paper, we primarily look at dysplastic repair as the chronic persistence of these KRT8 high transitional cells and these KRT5 cells. And so switching gears into our data in itself, we were very fortunate to work with Dr. Peter Chen and his group over at Cedars-Sinai, where they had enrolled individuals who had developed lung fibrosis after COVID that was so severe that they needed a lung transplant. I hope you can appreciate here, based on the Mason's trichrome staining, how abundant the collagen deposition is in these lungs. And so we did a very preliminary analysis in these lungs in terms of the alveolar epithelium itself. And you can see, compared to a control lung, these PASC fibrosis lungs exhibit this dramatic sustained reduction in AT1 and AT2 cells, which had been reported by several other groups at the time as well. In addition to that, we wanted to take a look at some of these players of interest, so the CD8 cells and those KRT5 and KRT8 cells that I just mentioned. And you can see how in a control lung, for the most part, you really don't see an appreciable representation of these cells at homeostasis. But I hope you can appreciate in these PASC fibrosis lungs, you actually see a large amount of these KRT5 and KRT8 cells, as well as abundant infiltration of CD8 T cells in these areas. And so this had also been reported by other groups, including us at the time. And this wasn't terribly surprising either, because these were people that had literally reached end-stage fibrosis to the extent that they needed a lung transplant. So the fact that there was so much fibrosis and there were hallmarks of dysplastic repair was not too surprising. But what did surprise us was when we looked at the distribution of the CD8 T cells themselves. We found that these cells primarily seemed to cluster or localize in and around these areas of dysplastic repair, as it's quantified here. And this seemed to be a unique phenomenon of post-COVID pulmonary fibrosis. You can see how in acute COVID, you don't really see this association, which we think is because there's just so much inflammation and abundant CD8 T cell infiltration at the point where it's the vast majority of the lung is infiltrated by these cells. We also were able to get a couple of lungs that had developed fibrosis after influenza infection, and we see a similar phenomenon. But perhaps most interestingly, we do not see this in other fibrotic conditions such as IPF. So this suggested to us that perhaps this association that we're seeing is a unique feature of viral infection-mediated fibrosis rather than a pan-fibrosis phenomenon. And so we had identified the spatial association between CD8 T cells and these dysplastic progenitors, but we still didn't really understand what these cells were doing. And so we wanted to move this into a mouse model so that we could manipulate the system more to understand their role exactly. And so we used first a mouse-adapted strain of SARS-CoV-2, MA10, and this is because wild-type SARS-CoV-2 cannot productively infect wild-type mice. And we use both young and aged mice. I'm just showing the aged mouse data here because they do have a higher propensity to develop fibrosis. And here is an aged Black 6 mouse lung, 35 days post-infection. And I hope you can appreciate that we don't really see substantial persistence of these CD8 T cells or inflammation per se. And also, we don't see large areas of these lungs exhibiting those KRT5 or KRT8 signatures that we saw in the human lungs. And this is reflected when we tried to score them for fibrosis using a modified Ashcroft score as well. We also tried to use a BALP-C system or a model. And interestingly, we do see some persistent immune cell infiltrate, but that they do not seem to be CD8 T cell dominant. We're not sure. This might be a strain-specific distinction or perhaps something else altogether. But again, not many CD8 T cells, and at least when looked at by protein expression via immunostaining, again, we do not see these KRT5 or 8 cells, and neither do we see fibrosis. And finally, we wondered if this was an artifact of the mouse adaptation process. So we also used a clinical isolate, but used instead a transgenic mouse where they express the human ACE2 receptor. And once again, we don't really see any appreciable persistent pathology in these mice. So we were pretty disappointed by this, and we were trying to understand what might potentially be the cause for this. And while we don't know for sure, one hypothesis that we have is that when we looked at the dynamics of epithelial damage and regeneration post-infection, both in the Black 6 as well as the BALP-C mice, we see that while AT2 cells were quite profoundly affected during that acute phase and then later regenerated, the AT1 cell compartment was largely unaffected. And we wondered, based on other studies, especially a recent one that had come from the Tata group over at Duke, whether this requirement for AT1 cell loss to develop these transitional states is what is hindering the development of chronic pathology in these mice. But this is something that we need to work out specifically to see if that's the case. But in the meantime, we still wanted to model this phenomenon that we were seeing in humans. And so we used an alternative model that we're quite familiar with, which was the influenza pneumonia model. So we used the PR8 strain of virus. And we see here that now in young mice, I hope you can appreciate these areas that express KRT5 and KRT8, as well as have CD8 T cell infiltration in and around them. Now, while we see this in young mice, it's substantially higher in aged mice, as quantified over here, both in terms of the quantitative increase at this time point, but also the persistence of these hallmarks of dysplastic repair, as well as these CD8 T cells. And going back to that previous point about this differential AT1 cell loss, you can see here just a comparison of our PR8 and MA10 infection models, and you can see how, compared to the naive mice, you do see a loss of AT1 cells during the acute phase of infection, whereas AT1 cells in the MA10 model remain relatively unaffected. And we now wanted to take a closer look at that spatial association between the CD8s and the KRT8 cells to look at that immune epithelial association. And we see kind of similar to what we saw in humans, the acute phase of infection, there's not really a very strong correlation, which again, we think is because these lungs are so inflamed at that time point with all these effector T cells infiltrating into the lungs. But when you look at a more post-acute phase, around 30 days post-infection, when a vast majority of the T cells have contracted, you see that now the remaining T cells tend to localize specifically around these KRT5 and KRT8 areas. And this becomes even stronger of an association as you wait even longer, up to 60 days post-infection. And so now that we had a model where we could actually phenocopy this, at least this association that we were seeing in humans, we went back to that original question of what these CD8 T cells were doing in this space. And so we just tried a very simple method of first trying to get rid of them and seeing what happened. But given the integral nature of their activity to an antiviral response and viral clearance, we couldn't just deplete them from the get-go. So we chose to use an antibody-based depletion strategy where 21 days post-infection, we treated them with an anti-CD8 depleting antibody. And then, and this time point was chosen because we know for a fact that infectious virus has been cleared by then. And then we wait again until our chronic 60-day time point and then collect the lungs to see what has happened. So here you can see the control isotype lung, which looks very similar to the aged lung that I showed previously. I'm sorry, the lights have gone up. There we go. Yeah, you can see how this control lung looks quite similar to the H lung I showed previously. But then when I deplete these CD8 T cells, we not only lose those CD8s, but we also see a dramatic reduction in these KRT5 and KRT8 areas. And this almost suggested to us that these CD8 cells were somehow maintaining these dysplastic progenitors for an extended period of time. On the flip side, we wanted to look at the functional epithelial, the AT1s and AT2s. And you can see here again, compared to the control lungs, when we deplete CD8 T cells, we're actually able to restore or at least augment the levels of AT1 and AT2 cells, suggesting that depletion of these cells does indeed help alveolar regeneration. So this was very exciting to us, but we still did not fully understand what these cells were doing. And so to understand what might be happening in these different areas of the lung, we chose to perform spatial transcriptomics, which we did both in the human PASC fibrosis lungs as well as the mouse influenza lungs. And we essentially tried to look at the signaling pathways that might be differentially regulated within these areas of pathology with KRT5 and KRT8 versus the healthy alveolar epithelium that did not have those dysplastic signatures. And one of the strongest signatures that we saw that was conserved between both the humans and mice was an enrichment of IL-1 beta signaling and inflammasome within these areas. And this was very exciting to us because it's consistent with previous data, both from Bridget Hogan's group as well as Joo-Hyun Lee's group, who had shown a role for IL-1 beta in epithelial repair, and also had shown that, in fact, chronic IL-1 beta signaling could impair the differentiation of these KRT8 cells. So to see if IL-1 beta indeed is relevant to our system, we went back to our CD8 depletion model where we saw such a profound effect for this with regards to alveolar regeneration. And we looked at what happened to IL-1 beta post-depletion, and we see that there was a decrease in the bowel fluid IL-1 beta levels, suggesting that CD8s was somehow influencing the release of IL-1 beta in the alveolar space. When we looked at who the cells that were directly producing IL-1 beta, they seemed to primarily be CD64 positive macrophages, which made us think that perhaps the CD8s are then signaling to the macrophages to release this IL-1 beta cytokine. And so we were trying to look for signals that could explain this, and that might be derived from the CD8 cells to potentially stimulate macrophages. And we saw that these CD8s were expressing high levels of interferon gamma and TNF for an extended period of time. This is data from the PASC fibrosis lungs, but we also have similar data in our mouse lungs. And so we tried to block these cytokines in mice using a very similar strategy where 21 days post-infection gave them neutralizing antibodies towards the cytokine, and again, we're able to decrease IL-1 beta. So this suggested to us that perhaps these cytokines indeed are relevant for this process of IL-1 beta release, and they're potentially derived from CD8s. And just to I guess, complete the picture of the spatial localization, we also went back to see where these IL-1 beta producers, where these macrophages were. And again, this is just data from the human lungs, but we see the same pattern in mice, where they primarily seem to be distributed within these KRT8 high areas, as you can see here, kind of similar to what we saw with the CD8s as well. And so, although all our data so far, at least in terms of association, suggested that CD8s were possibly signaling to macrophages, we didn't really have direct evidence of this. And so we performed a somewhat reductionist experiment to test this. So we collected CD8 cells and macrophages from the post-viral lung and cultured them ex vivo, co-cultured them ex vivo to look at the release of IL-1 beta. And so here on the right is just IL-1 beta levels, the basal release into the supernatant when macrophages alone are cultured. And you can see how we see a pretty big increase when co-cultured with CD8 T cells, suggesting that indeed there is a direct interaction between these cell types to promote IL-1 beta release. And then, but when we add a neutralizing cytokine to either interferon gamma or TNF, we see that that increase is actually diminished, suggesting that these cytokines are indeed what are stimulating these macrophages to produce IL-1 beta. And when we block both cytokines, we in fact see an even more dramatic decrease in IL-1 beta. So we think there might be like a synergistic role for the activity of both of these cytokines to stimulate macrophages to release IL-1 beta. And so this was the model that we had based on the data that we'd generated at the time, where we think it was these persistent CD8 T cells that are chronically producing interferon gamma and TNF to stimulate this macrophages and monocytes to produce IL-1 beta chronically. And we think it's that IL-1 beta that is preventing the differentiation of these epithelial progenitors. Although the CD8 depletion data was very remarkable, we recognized that that was not really a clinically viable strategy to treat this condition. And so we wondered if perhaps blocking these cytokines might be a way to confer the same benefits. And so we tried blocking interferon gamma and TNF, the same strategy as I described previously, where 21 days post-infection of these mice, we treated them with this neutralizing antibody towards these cytokines. And when we looked at the lungs of these mice, you can see these control lungs, again, have lots of KRT5, KRT8, a decrease in AT1 and AT2 cells, but that upon treatment seems to be rescued, where we're both seeing this decrease in KRT5 and KRT8 cells, as well as an increase in AT1 and AT2 cells, suggesting that indeed this could be therapeutically potentially used to boost alveolar regeneration. And we worked our way further down this cascade and explored the possibility of neutralizing IL-1 beta to potentially rescue this phenotype. Again, very similar strategy, 21 days post-infection, use blocked IL-1 beta instead. And we're able to phenocopy exactly what we found with the CD8 depletion, as well as with the interferon TNF blockade, where we see both a decrease in the KRT5 and KRT8 cells, as well as an increase in AT1 and AT2 cells. So while this was very exciting, again, we didn't have direct evidence that it was IL-1 beta specifically that was preventing this differentiation process. So we adopted a 2D AT2 culture system that we'd actually gotten from Dr. Rachel Zemans, where instead of just media, we instead used conditioned media from that ex vivo culture system that I mentioned previously. So we essentially wanted to see what it is about the interaction of CD8s and macrophages that was influencing AT2, but in a more controlled system. And with that, we then looked at the expression of transitional cell markers or AT1 cell markers to use that as a surrogate of the differentiation process. And so there's a lot going on here, but I can walk you through it. So if you see, these are just the different markers, gene expression of different markers that are expressed by transitional cells. And in blue are AT2 cells that received conditioned media from macrophages alone. And you can see how it's somewhat comparable to the levels of AT2s receiving regular media. But then when we use conditioned media that was derived from that macrophage CD8 T cell co-culture, there's actually a pretty big increase in the gene expression of these transitional cell markers. But to that same conditioned media, if we added a neutralizing antibody to IL-1 beta, you see how these expression levels are greatly diminished, suggesting that it's specifically IL-1 beta in that conditioned media that's driving this transitional cell marker expression. Below is just the converse, where we looked at AT1 cell markers, and you'll see the gene expression patterns are just reversed, wherein you see higher AT1 expression levels, AT1 marker expression levels when treated with media from macrophages alone, but that is diminished when you get media from CD8s plus macrophage, and that's rescued again when IL-1 beta is neutralized. So with that, we really do think it is particularly IL-1 beta from this system that is driving this phenomenon. And to see whether this extended into humans and IL-1 beta was relevant in that context, we actually used a different cohort of COVID-19 convalescents that had been enrolled at UVA, and we stratified them based on their lung function post-recovery. And you can see how individuals that still maintained abnormal lung function were the ones that had significantly higher circulating IL-1 beta levels than those that had normal lung function. And this has since been replicated across other studies as well. But I'm highlighting one where they sampled the bowel fluid and see a very similar trend where IL-1 beta was elevated in patients with impaired lung function. And in addition to all of those cellular changes that I mentioned, I also wanted to make a point that this was reflected in the lung function of these mice as well, post-intervention. So be it CD8 depletion, interferon TNF blockade, or IL-1 beta blockade. I'm just showing the static compliance data here, but we actually see this pattern across several metrics where after treatment, you can restore lung function to levels comparable to naive mice, suggesting that this does indeed confer physiological benefit in these mice. And so with that, I will summarize by saying that we were able to use numerous cohorts of PASC patient human samples, and were able to develop a mouse model of post-viral fibrosis, which really helped us identify this aberrant immune epithelial progenitor niche. And essentially, this niche is composed of CD8 T cells that are excessively producing IL-1 beta, or sorry, interferon gamma and TNF to stimulate these monocyte-derived macrophages to secrete IL-1 beta chronically. And we think that in turn is signaling to the epithelium and preventing successful alveolar regeneration. And while we use a variety of experimental treatments where we either deplete CD8s or neutralize these cytokines, we think there might be potential for existing FDA-approved therapeutics that are targeting similar signaling cascades to potentially rescue this phenomenon and break that loop to allow functional regeneration to occur. And so with that, I would like to thank my mentor, Dr. Ji Sun, for all of his help in making this happen, members of the Sun Lab, particularly Insu and Wei, as well as collaborators at Cedars-Sinai, particularly Dr. Peter Chen, Dr. Tanya Luck, and also at UVA, Sean and Dr. Changji Zhang, who without whom this work would not have been possible. And I'm happy to take any questions.
All right. Thanks so much, Harsh. That was fantastic. I know I elevated you to doctor at the beginning, but I think you'll have no problem with your defense. Just really good work.
Appreciate that.
So before we get to the discussion portion, I just wanted to remind all the listeners that please feel free to put in any questions in the chat. I don't know if I see any just yet, but as the discussion goes, if you'd like to post something, we will try to get to it. So to start the discussion portion, I think I'll just ask a fairly low-hanging question, which is what causes these CD8s to be exuberant? You know, what's triggering them to be kind of this chronic activation? And I know it's likely speculative, but you know, what would cause this type of CD8 activity in the lungs?
Yeah, no, that's a fantastic question. You say low hanging, but that's possibly the most important one that we actually have not answered. And like you said, we still don't know for sure, so it's speculation at the moment. But so one thing we did look for, because there was a pretty active idea in the field that perhaps there are vital remnants or vital antigens of some sort that are possibly triggering this, that stay in these individuals and are triggering this. We looked in all of our lung samples, both by immunostaining for vital antigen, but also or protein antigen, but also RNA scope, looking for vital RNA. And we really didn't find any evidence of this in our human samples. We did the same in our mice, and at the time points that we were looking, again, we did not really see this. So this could, I don't think that excludes the possibility of there being vital remnants. It could just be a technical, limit of detection problem. So that is a possibility. But other than that, I think there's the other possibility of just some form of an autoimmune response that's being activated after that initial damage. So potential antigens, presumably autoantigens that are being expressed either by the epithelium or other cell types within that vicinity that are stimulating these cells. The other hypothesis is perhaps there's also just signals that are keeping these CD8 T cells in there. So like TGF beta, for example, is something that's also known to be elevated with aging. And interestingly, some of these cell types that I described, the KRT8 cells, are high expressors of the integrin that cleaves TGF beta. So when you have these sources of active TGF beta in the vicinity, perhaps that is in part at least recruiting them and keeping them there to activate them further. But again, like I said, this is all speculation. We don't really know yet. One of the first questions we're trying to answer is whether these T cells are vital specific in the first place. I feel like that would be a clue to begin with. We haven't been able to answer that in our human lungs. In our mouse lungs, we definitely see a large number of them. But again, does that directly mean that that's vital antigen that's activating them at the time? It's unclear. Sorry, that was not a very satisfactory answer, but that's the
No,
best
no,
I've
no,
done.
that was great. I know it's an obvious next question, but absolutely not an easy question to answer for sure. You did cover another question of mine, which was about the specificity of these T cells, but I'd like to bring in Angela because I think I saw her virtual hand raised. So take it away.
Thank you so much. And this, you may have partly answered my question already, but I'll ask it anyway. I get one question I had was what we know about these lungs, about these human lungs that we got, and how long after their acute infection were they? I'll say as an ICU doctor, the patients I saw needing transplant largely sort of had horrible ARDS and then it just kept going. And, finally, months later, it's like that's completely fibrotic. They have to go to transplant. I did not see many people, in contrast, in my outpatient clinic who had some fibrosis. Their PFTs took a hit, but they didn't tend to go to transplant. So my first question for you is, do you think this is partly an ARDS signature and not so specific for viral? And I guess that gets a little bit into the CD8 question. Have you looked at any non-viral, sort of post-infectious, post-ARDS lungs to see if there's a similar pattern?
Uh, it's a fantastic question. So to answer your second question first, no, we have not looked at any non-viral. The most we've looked at in our human samples is influenza and SARS-CoV-2, both acute and later phases, but otherwise have looked at non-viral like IPF, but beyond those categories, we really have not explored it. I think it's a very interesting question. But yeah, to go back to your first question, at least in terms of the time points that we were getting these samples, it varied significantly, but went from anywhere from 1 to 2 years post-acute disease that these people needed a transplant. They were not hospitalized throughout the duration of this phase or until they got the transplant. So I think purely based on them the severity of the acute disease, I think it's impossible to discount ARDS as being a driving factor in this phenomenon. But I think what your second question was in terms of
Yeah,
looking at non-vital ARDS, I think that would be a very interesting control to like delineate the respective contributions of these,
yeah.
which, yeah, I think that would be a very cool thing to pursue.
But I agree with you. 2 years would be a long time to sit in sit in
Yeah,
ICU with
yeah, yeah,
ARDS. So
yeah.
there's probably some mix of patients. So thank you
Right.
so much.
Thank you.
I think I saw Charles' hand up as well.
Yes, congratulations for your presentation in the paper. I have
Thank
two
you.
questions. One is you kind of nicely show in your paper that you need the TNF and interferon gamma blockade as well as the IL-1 beta blockade and give you the decrease in pathology. I guess clinically, the TNF alpha blocker really wasn't working, right, in terms of protecting severe disease. Do you think that's the reason why it didn't work, that you need this combination of interferon gamma plus TNF blockade clinically? And I,
Sorry, go ahead.
and I had a second question, but go ahead.
Okay, okay. Yeah, I think one distinction, and it becomes hard, I'm sure, I mean, much harder than I can appreciate, is the timing of it all and when you transition from that acute COVID
Mhm.
phase of infection to this post-COVID fibrotic phase of disease. So at least in our data, we think all of these cytokines that we're blocking at this acute phase are absolutely essential during that acute phase to both allow the inflammatory response to like work to its fullest capacity to clear a virus, but also um, in terms of epithelial repair, things like TNF and IL-1 beta have actually been shown to be important to start that
Mhm.
cascade of regeneration as well. We just think there has to come a stop where after the viral clearance, after that initial insult, as the immune response should largely contract, there seem to be some facets of it that for some reason does not. And that goes back to the first question. And I think at that phase, using these, say, potentially even TNF alone, but at least based on our mouse data, but maybe we've not actually explored in our in vivo setting using just TNF blockade. But I would imagine, based on our in vitro or ex vivo system, that there should be some benefit. But I think the biggest determinant there seems to be the timing of when to
Mhm.
treat or neutralize these cytokines.
My other quick question is, you guys actually show that the CD8 alpha in this viral model correlate quite well with the KRT sort of responses, but not in IPF. Do you think this is because the IPF samples are more sort of the downstream advanced disease? It takes a while, if you were going to do this again in 10 years and repeat the study on the same patients, would you lose that signal?
Yeah, that's a very interesting question, and I honestly don't know. It's something we've floated as, is this like an interim or like an early phase of what could become full-blown IPF that we're capturing with this system? And we honestly don't know. Our initial inclination was that it's all about that vital type 1 kind of inflammation that is set up after infection that seems to set the stage for this large scale CD8 biased response. But I don't know for sure, or I don't even know how to begin to speculate as to how that might potentially change at a more later protracted time point when they potentially go through like full blown IPF at the time. But it is a very interesting question. I mean, this does not answer your question, but just as a point to add, we do see elevated CD8s even in IPF patients. It's just we don't see that spatial association like we do with the viral infection. So it's not at the same magnitude as the ones post-viral infection, which I guess is to be expected. But yeah, I think it's an interesting question that we don't really know yet.
Thank you.
Could I chime in?
Yep.
Yeah, Charles, it's a great question. More recently, with collaboration with Peter's group, looking at the mild IPF versus really severe terminal IPF. In the really severe terminal IPF seems more really the traditional TGF beta collagen some kind of type 2 response. But with the mild one, maybe represent the early development. Actually, the gene set with all interferon antiviral response actually came up. So suggesting maybe there's as you suggested, maybe there's kind of a transition from initial development, more maybe viral or other kind of inflammatory mediated injury to later when it become terminal IPF is more like the traditional. But the point is the window to target probably is early, right? Once you have so much of collagen, maybe it's harder really to target. But it's a great, fantastic question.
Thank you very much.
I'm gonna pick up on that question of timing and move to a question that we have posted by Scott Randall in the chat. So Scott says, interestingly, IL-1 beta is added to early human AT2 cultures and supports growth. Could IL-1 beta inhibition be harmful in early infection or repair processes?
I think Harish partly answered this question already.
Yeah.
Sorry, go ahead.
Yeah. Well, if you want to add anything more to that, that would be fine. But I do have a second question.
I mean, I guess I could just add that while we did not test that specifically in our experiments, we just based on some work from Bridget Hogan's group and also what we think of in the early acute phase of infection from like the immune standpoint of view, I think you're completely right in that IL-1 beta inhibition at that early time point would both hamper the immune response, but also hamper the regenerative response in the alveolus. But please, I'd love to hear your second question too.
Yeah, so I think it's this underlying big picture question about folks that end up with PASC or long COVID. They were on the path towards pulmonary had subclinical pulmonary fibrosis, or were on the path towards pulmonary fibrosis, and this infection sort of pulled it out of their background. And how many of those folks had the MUC5B polymorphism or TERT abnormalities that would predispose them to this kind of chronic lung pathology?
Yeah, that's a fantastic point. And it's one that, we really can't delineate in our study, but I think it's a very important question of how much of this just exacerbation of underlying propensity to develop fibrosis. And we're independently testing that in some mouse models as just viral infection mediated, like post-bleo or something like that, to see what would happen. But it doesn't necessarily answer the exact same question that you're asking. And to be honest, I don't know beyond, like, say, looking at the genetic predisposition to developing IPF or risk factors in that regard, unless we had a longitudinal imaging cohort, something like perhaps they have in the UK Biobank or something like that. I don't know if we would be able to capture that delta from their baseline pre-infection to post. But I think that's a very, very interesting question, in that were they all destined for fibrosis anyway?
The excellent graduate study progress and a wonderful presentation. Thank you very much.
Thank you so much.
I see Jay's hand has been raised.
Thanks. I also want to start by saying congratulations. It's really beautiful work to the whole team. Congratulations. It's generated a lot of ideas and provocative thoughts. I guess I'd like to encourage thinking about what are the CD8 T cells doing exactly? And you sort of show that when you get rid of the CD8 T cells, everything disappears. And you show that there's sources of interferon gamma and TNF alpha. And if you block those two things, everything disappears. But, I would guess there's other sources of interferon gamma and TNF alpha, like CD4 T cells, and there's lots of activated T cells and CD4 cells and B cells in these post-viral lungs. And so I guess, and then you also show that type 1 cell death seems to be really strongly associated with whether or not you get this. And of course, we think of cytotoxic T cells as providing perforin granzyme and killing cells. And so do CD8 T cells have really specific activities that they're doing that only they can provide? And are, for example, the activated CD4 T cells in these lungs, are they downstream of CD8s, or are they not downstream of CD8s? Is the type 1 cell death downstream of CD8s or not downstream of CD8s? Or what specifically do you think is unique to the CD8 T cells in this story?
Yeah, that's a very interesting question. Again, speculation, I'll provide the disclaimer to begin with, but I think one thing is the anatomical distribution of the CD8s within the alveolar space. When we look at CD8, CD4s, B cells, and the general distribution across this, the CD8 seems to be the most abundant in the parenchyma of these lungs relative to CD4s and B cells, which, although you definitely do see them in the lung parenchyma, you tend to find them more in those, organized clusters, the eyeballs or TLOs, depending on what you'd like to call it. But I think one is just the proximity to this space where they're able to exert their function. The point about directly causing AT1 cell death is very interesting. It's something we considered very early on to potentially look at, say, like an MHC1 inhibition or blockade and see if you're able to rescue that. It's not something we pursued, so I don't know. I couldn't tell you with any certainty what that would look like, but I do imagine that having these activated cells, if it also comes down to what the antigen of choice is here that is driving this response. So if it is something being presented by the epithelium, I could hypothesize a scenario where they're actively causing the damage in there. But beyond that, I think it's just like you said, they're not the sole source of gamma or TNF, but they're both in terms of proximity and also quantity amongst the most prolific producers in that area. And I think that seems to contribute to the observed phenotype. And there was something else, another point you mentioned that I meant to come back to, and I cannot remember right now. But I hope that somewhat answered the question that you asked.
It does very much. I guess maybe I'm not sure if this is the point you were thinking of, but do you know when you deplete the CD8 T cells, did you affect the CD4 T cells and the B cells in those lungs?
We did actually. So we had a previous study where we looked at the CD8s and their role in chronic pathology in the influenza model. Nick Goplin, who I believe you've met as well. But yeah, he had actually shown that when you deplete the CD8s, you actually end up manipulating a variety of cells in their CD4s, B cells. Interestingly, in our study, we also saw the macrophages, the same ones that we think are the producers, they also go down in number. So, and looking at some RNA-seq data, we see evidence that the CD8 themselves might be producing some chemokines that could serve as macrophage attractants as well. So we think they definitely play a very central role in this process. And we don't necessarily think depleting CD4s or CD8s would confer the same effect in like as a master regulator in this context.
I think that's a nice lead into a question we have in the chat. So I think you really beautifully showed the removal of CD8s, ameliorating the disease. But we have a question from Christy Nelson who asks, can you transfer these pathogenic CD8s to new hosts and do they cause disease?
Yeah, that's an excellent question. Were you a reviewer, maybe? It was actually something that we were asked to do, and we were slightly hesitant before performing the experiment because G and I had discussed it before, and one concern that he had, which completely made sense to me, was the fact that these cells are so activated at the time that transferring them into this new host could potentially just, end up killing them, and you don't necessarily see their function. But regardless, when we were asked to do it, we tried to do it, and we didn't exactly do. I guess, sorry, I'm getting ahead of myself. So the way we designed this experiment was we infected aged mice, and at that 21 days time point when we were depleting CD8s, we instead isolated CD8s from the lungs. We enriched for them, and then we intratracheally transferred them into young or aged recipients. And the question being, is this an age-specific phenomenon, and can you transfer these age potentially pathological T cells into these young hosts to cause disease? And the results were a little mixed. And one of the issues is that we don't know how much of that is an artifact of T cell viability in this context. But what we saw is, I think, if I remember correctly, AT2 cells were diminished relative to, a young recipient that got young T cells. Whereas when we looked at these dysplastic cell markers, we really did not see a difference. So it suggested that there was some level of pathology that could be transferred by just moving these T cells from one host to the other, but it did not fully phenocopy what we were seeing in like a truly in the same environment. The alternative explanation is that there's something about the lung as well, like the lung environment, that makes them less resilient to repair themselves and sustain this kind of damage. So I guess to Christine's question, we didn't exactly do the experiment that you're suggesting, but it was more of a heterochronic transfer to see if there was an age-dependent effect of this. And it does seem like there is one, but it's not solely dependent on the CD8 T cells.
Christine says thanks in the chat. Continuing with our theme of questions about the animal model itself. So Amali is asking in the chat, in your animal model, have you reinfected mice after the cytokine blockade? I wonder if the AT cell that repair in this way are functionally normal.
That's a great question. I don't think we have a G. I don't know if you can chime in based on anyone else's work, but at least in our study, we did not really explore the idea of reinfection after treating. One hypothesis, which is, again, it's a tangent from the question, is that if we can artificially, like we went after the question of why this occurs, why do we have these CD8s that persist in these areas for an extended period of time? And one hypothesis that we had is that this is like supposed to be an evolutionary mechanism where these vulnerable areas of the lung, which are undergoing ongoing repair, are secreting these chemoattractant molecules to keep these CD8s as rapid defenders right in those sites in case of subsequent infection. So one of our questions at one point we had thought about is, if you manipulate this environment to get rid of these pathological T cells, and then you reinfected them, will they do worse than a mouse that you did not manipulate in this way? So I guess we were looking at it more of an immune-centric point of view, in that is there a reason for these memory T cells to be here? But I think your question about the epithelium itself and how it might be modulated by these different cytokine interventions is actually something we did not really consider. again, I'll open it to G if he has anything to add.
No, that's a great question. We and also great answer from you. We don't have anything else to add, but it's a good question. We should think about in the future.
Can I ask a follow-up question related to that? I'm sorry, Charles, unless you were going to follow up on the same one. I don't want to. Okay, that's a really interesting question by Amelie, and also not only does it bring up the question about whether the epithelial cells have changed and might alter things, but also now you've got memory CD8 T cells, and it might help you to differentiate autoimmunity versus antigenicity. You showed SARS-CoV-2 didn't induce this sort of a PASC phenotype, but maybe in a mouse that has recovered from influenza, if that is an autoimmune CD8 T cell now after in SARS-CoV-2 or other sorts of injury, maybe you'd get this autoimmune response, and you would. It's a really interesting suggestion, I think. And I don't. It's not really a question as much as a follow-up on. In addition to following the epithelial cells, I'd be interested about the CD8 T cell memory in those mice.
Yeah, 100%.
Yes, I mean, sort of related to that, I was just curious, in your aging animal studies between the flu and SARS-CoV-2, you didn't see any sort of chronic changes with the SARS-CoV-2 in the aged mice, which kind of interesting. And so, do you have any additional insights? Is it the virus, or because even for bleomycin study, they are pushing for using older mice for the bleomycin response. I'm just curious why the differential viral response.
I'm sorry, can you I missed. So are you asking me why the differential viral response in the SARS-CoV-2 model specifically, or
no, in the age animal versus young,
okay,
flu
okay.
versus COVID, SARS.
Yeah. So I mean, just first touch on the COVID. I think regardless, so we used aged mice based on our experience with influenza, where we had reported and consistently see both this exaggerated immune response, but more alveolar injury, and then this protracted recovery response. And again, we don't specifically know what it is about the aged environment. Is it the T cells? Is it the epithelium or the lung microenvironment in general? Again, that transfer experiment that I was describing a little bit kind of alludes to the idea that the T cells, at least in part, contribute to this. There's something about an intrinsic change to an aged T cell that could be contributing to this, but it does not explain the phenotype fully. One alternative idea is we found that these aged lungs also have higher levels of TGF-beta, which again can affect the epithelium, but also the T cells themselves. And we think that in terms of TRM differentiation and function, it could be acting in that way, but also towards the epithelium. Every part of the lung microenvironment, the epithelium, the stroma, and the endothelium could be affected by excessive TGF signaling. We don't know for sure if that is specifically the reason why we see this effect. But with the COVID model, we hoped that the aged mice would kind of recapitulate what we see with flu, but both young or aged mice, it did not matter. We, again, just didn't see that AT1 cell loss. And as for now, that is really the only idea we have as to why that system is not working as well as the flu model. So we're thinking of ways to potentially artificially induce AT1 damage and see if that could mimic things that we're seeing with flu. But at the moment, at least, we don't know for sure.
Thank you.
Thank you.
I don't see any more hands, so I'll ask a quick question. I apologize if I missed this, but the initial human data looked at PASC post SARS-CoV-2, the model looking at influenza. Did you have, or are you interested in looking at possibly human samples from H7N9 infections? Do you see these hotspots and the same sort of localization of macrophage and CD8s?
Yeah. Again, are you a reviewer? No, because it's an excellent point, and I think it would also expand our findings, I think, to PASC PF or COVID specific phenomenon to something potentially being relevant to any viral infection that can cause parenchymal abnormalities. So we've not looked at 8, 7, and 9 specifically, but we are interested in looking at more of these post-flu samples. I think the biggest issue thus far has been trying to acquire said samples because a large amount of, I don't know about 8, 7, and 9 specifically, but at least the large amount of samples that we were able to access are either from that acute phase of infection or are confounded by a potential bacterial secondary pneumonia. And I think that makes interpretation a little challenging. How much of it is the original infection versus the secondary infection? But I think it's a very important question. Currently, I think there's just not as much attention on the post-flu complications long term as much as there is for COVID. And I think that's why this has kind of flown under the radar. Again, this is further confounded by a lot of these samples, for example, were collected between that 2021 to 2022 phase, like early vaccine rollout period. When it comes to flu, we have God knows how many prior exposures through just direct infection and vaccination and things like that. So how much of the memory is, a boost versus the de novo response? Again, those are potential confounders there. But I think it is an area I think that needs to be studied more because who knows what's out there next,
Absolutely. Long flu. That's the
Yeah, long flu, exactly.
All right, so we are at the top of the hour, so that brings us to the end of our journal club. Many thanks to our panelists, Dr. Sun, Rogers, De La Cruz, and Misgerd, along, of course, with Harish, our presenter. Thanks to everyone participating live online today. Remember that the journal club was recorded and will be posted on the ATS website, and please reach out to your section or assembly with future journal club suggestions. Okay, thank you so much, everyone.
Thank you
Thank
so
you.
much.
Thanks so much
Bye.
for organizing.
Thank you. Bye.